to introduce yourself? Sorry, before I do that, can you spell your name for the court reporter?
Yes, my name is Lisa, L-I-S-A. My last name is Ellison, E-L-L-I-S-O-N.
Can you tell the jury what it is that you do?
Yes, I am a forensic biologist, so that means that I look at evidence for the presence of bodily fluids such as blood and semen. I can also perform DNA analysis if necessary.
What sort of training or background do you have to be able to do this sort of work as a forensic biologist?
I received a Bachelor of Science degree from the University of Georgia in 1995, and that was in biology. Soon after that, I began working at the Georgia Bureau of Investigation, which is where the majority of my training occurred.
How long did you work for the GBI?
I worked for the GBI for approximately 11 years.
After leaving the GBI, what sort of work did you start doing?
After leaving the GBI, I started working at the Defense Forensic Science Center in May of 2006, basically doing the same thing. The Defense Forensic Science Center is the crime lab for the military, so now I work on cases that happen on military bases or are involved with military individuals.
You said now, so are you still doing that as of today?
Yes, ma'am.
Now, in addition to your bachelor's degree, you said you received most of your training from the GBI. Tell the jury what kind of training the GBI specifically does for their forensic biologists.
Okay. When I graduated from college, I started at the GBI in September of 1995. I was an intern there in the fingerprint section, and then I was a technician in the toxicology section. It wasn't until July of 1997 that I became a forensic biologist in the serology DNA branch of the laboratory. Once I started there, I was trained in serology, which is the identification of bodily fluids. At the time, we had tests for blood, semen, and saliva. When I started my training, I was assigned a primary trainer, and it was his job to explain to me the type of test to perform, as well as how to interpret the results. I went through several written and oral exams, which I passed successfully. I also was given a set of what we call practical samples, which I had to use my training to obtain 100% accuracy on the samples. My trainer knew the results, but I did not. I did obtain 100% accuracy on that test as well. Soon after that, I began doing serology analysis only on cases that came into the laboratory. And then a year later, I started DNA training. It followed basically the same idea. I had written exams, oral exams, which I passed successfully, and then at the end was given a set of samples in which I had to obtain 100% accuracy, and I did. Every year since I have been trained, I also have to perform DNA analysis on what's called a proficiency test. It's an outside company that sends in samples, and again, I have to obtain 100% accuracy, and I have done that approximately twice a year since I've been in the field in 1997.
So in 2019, most of us have heard of DNA, kind of had a basic understanding, but what is the difference between serology and DNA?
So serology is the identification of bodily fluids. So there are tests that I can perform to say that something is semen or something is saliva or blood. DNA analysis is actually taking that testing a step further and looking to see if I can determine who the donor of those fluids may be. All of those fluids have DNA in them, so I can perform DNA analysis on those fluids and go from there.
Is there any way to leave your DNA behind on a particular piece of evidence without leaving fluids, as you just discussed?
Yes, ma'am.
Can you tell us what that is?
Typically, it's through touch.
Okay. Now, throughout the course of your career with both the GBI and as with the Army, Department of Defense, have you had an occasion to test items for what we call touch DNA?
Yes, ma'am.
Any way for you to estimate for us how many items you've tested for touch DNA over the years?
I mean, I can't give you an accurate estimate, but I would say at least 1,000. Most of that was with the Defense Forensic Science Center.
Have you ever had an occasion to testify as an expert in the field of forensic biology?
Yes, ma'am.
And any idea how many times you testified as an expert in the field of forensic biology?
Approximately 175.
Judge, at this time we would tender as an expert in the field of forensic biology.
We would agree that she's an expert.
All right, she's admitted.
Back in 2005, where were you working?
I was working at the Georgia Bureau of Investigation in the Division of Forensic Sciences, or the GBI Crime Lab.
Did you have an occasion to work on any items that related to the disappearance of a female by the name of Tara Grinstead?
Yes, ma'am, I did.
Specifically, what was the first piece of evidence that you looked at in this case? Do you recall?
The first piece of evidence that I tested was a portion of a latex glove.
Show opposing counsel, it's been previously marked as State's Exhibits 28, 29, and 91. What that's supposed to say. I'll change it. May I approach the witness?
You may.
I'm going to show you what's been previously marked as State's Exhibit 28, 29, and 91. Ask you to take a look at those for me, please. I know it's been a while since you worked for the GBI, but the GBI had a certain protocol as to how evidence was to be maintained before it came to the crime lab.
When evidence was submitted to the crime lab, it was required to be in a sealed condition.
If it was not sealed appropriately, would that have been indicated on a report related to the item?
There would have been some notation, I'm not sure exactly back then how it would have been done, but there would have been either notes in our laboratory system or a report would have been generated to that effect.
Does the GBI crime lab also assign its own case number as it relates to each case that y'all test items for?
Yes, ma'am.
And would taking a look at one of your reports refresh your recollection on what the case number was for this particular case?
I actually know the case number.
Well, I'm impressed. Okay, so tell us what that case number is.
It's 2005-10257070.
Now, looking at the exhibits that you have in front of you, that being State's Exhibits 28, 29, and 91, can you tell us if the case number that is indicated on those exhibits matches the case number that was used for the items related to the disappearance of Tara Grinstead?
Yes, it's the same case number.
And based on that, are you able to tell us whether or not that is in fact, those are in fact pictures of the glove that you tested?
Yes, it appears to be.
Your Honor, this time I would tender into evidence State's Exhibits 28, 29, and 91.
Any objection?
No, sir.
All right, they're admitted.
Take those from me. Mr. Rigby has better luck with this quickly than I do. So we'll start with State's Exhibit 29. Now, looking at stage 29, to be clear, did you take this picture yourself?
I did not.
However, this particular picture, is that consistent, I guess the scale that we see there, is that something that's commonly used to show the actual size of items?
Yes, ma'am.
And then State's Exhibit 91, which has also been tendered. Again, would that be that same glove, just without the scale?
Yes, it has the same case number attached to it.
Now, you mentioned earlier that you tested a portion of the glove. Can you tell us why it was that you tested only a portion of this particular glove?
In this instance, I believe that this particular item, being the glove, was going to have some other testing done on it, fingerprint analysis, for example. So I was given just a portion for DNA analysis that would not interfere with any latent print or fingerprint analysis.
So let me ask you, your knowledge now of touch DNA. If they attempt to lift fingerprints from something, can that remove the touch DNA?
It can. There are processes in place with latent print technology that we can try to limit that. But yes, the latent print testing can remove DNA.
And then the other way around, if you swab something for DNA, is the possibility you're going to mess up the print itself?
Yes, ma'am.
I'm going to show the jury what's been marked as stage 28. Can you tell me, looking at stage 28, which portions of this glove that you actually tested for DNA?
I actually tested the portion, it looks like the fingers of the glove had been cut, and then there's kind of an upside down U portion of the glove.
Yes, ma'am.
That's the portion that I tested, which is basically this part of the glove.
Okay. Now, when we say testing, can you tell us what you do to actually try and obtain a DNA sample from that portion of the glove?
So in this particular case, I took a swab. It's basically a cotton Q-tip type swab, wet it with some sterile water, and then I just rubbed it along the surface of that middle portion of the glove on both sides.
Once you do that swab, use the swab to obtain that, what process is used to see if there is in fact any human DNA on that swabbing?
When I perform DNA analysis, the first step is to actually take an item, and in this case, the swab that I took, treat it with chemicals. I put it into a tube first, treat it with chemicals to extract any DNA. There are several components to a cell, but the DNA is the portion that I'm concerned with. So this extraction process actually pulls the DNA out of the cells and cleans up the rest of the sample so that at the end of the extraction, I'm left with a tube that just has DNA in it. The next step is to see how much DNA I have. And after that, I then quantify it and then put it on a genetic analyzer. Back in 2005, the genetic analyzer would give me information at 13 areas of someone's DNA profile.
How many areas are there total?
Oh, there's thousands,
Okay.
tens of thousands, but we're looking at 13 areas where individuals can be unique or be different. Because we are all humans, 2 eyes, nose, mouth, much of our DNA is actually the same. So this is 13 areas where we can actually differ that we're looking at. That was done on the swab from the glove, and it can also be done on samples from individuals for comparison.
How do we normally obtain the sample from the individual for comparison?
Typically, if there's someone that law enforcement wants us to compare to something, DNA profile from a piece of evidence, it's either a blood sample or a swab from inside of their cheek.
Just a second.
You can go ahead.
What do we call the swabs from the cheek usually?
They're either called buccal or buccal swabs.
Is it spelled differently?
No, just depends on where you're from as to how you pronounce it.
Kind of like pecan and pecan?
Exactly.
Okay. So in looking at this particular sample, the swab that you did from that portion of this glove, was there any DNA, human DNA found?
Yes, ma'am.
And are you able to tell us if that was from one person or more than one person?
The DNA I obtained from the swab of the glove appeared to be from 2 individuals.
And again, because most of us watch shows like CSI and those sorts of things, we have an idea in our heads of what you're looking at. Let me ask you, are you able to tell anything identifying about the persons based on the DNA sample?
No, ma'am.
Are you, however, able to do a comparison to see if it matches someone's DNA?
Yes.
In this particular case, did you have any comparisons that were used to determine if Tara Grinstead's DNA existed on that glove?
I was given a toothbrush that supposedly belonged to her, and I performed DNA analysis on that toothbrush and developed a profile for comparison.
Okay, and to be clear, you said supposedly belonged to her. Do you have any reason
Yes.
to doubt that?
No.
Okay. But did you personally collect that toothbrush?
No, ma'am, I did not.
However, was that toothbrush used as a known reference sample for Tara Grinstead?
Yes, ma'am.
And when that toothbrush was compared to the DNA sample found on the piece of the glove, was there a match?
Yes, ma'am, there was.
Based on the information you received from the agents who collected the toothbrush and your work as a scientist, are you able to tell this jury whether or not Tara Grinstead's DNA was found on that glove?
Yes, I'm able to determine basically a frequency of occurrence of how rare her DNA profile is.
The other DNA sample that existed, were you able to tell whether it was a male, female, adult, anything of that nature?
I was able to determine that it was a male.
How do you determine that it's a male?
So I talked about the 13 areas that we look at on a DNA profile. There's actually a 14th that tells us what gender a DNA profile is. So I knew I had a mixture of 2 individuals, and I was able to determine that one of those profiles matched Tara Grinstead or the DNA profile from that toothbrush. But at that area where I can determine gender, there was a Y chromosome, and Y chromosomes are only found in males. So I deduced at that point that the other individual was a male.
Now, we've been talking about DNA, and I kind of want to clarify. In 2005, where did we obtain, for at least for criminal justice purposes, the majority of the DNA from items that were recovered by law enforcement?
Most of our evidence samples, we got DNA from the bodily fluids that I talked about, blood, semen, saliva.
Okay. This idea of touch DNA, was that a kind of science that was normal in 2005?
No, ma'am.
What about now in 2019?
It's predominantly the type of samples I test now.
And have you ever testified in court as it relates to touch DNA?
Yes, ma'am.
And have you, I know you say you testified mostly in military courts. Do you know if you've ever testified in Georgia courts about touch DNA?
Not that I can recall offhand, no, ma'am.
Touch DNA, and again, I know that that sounds very simple, but can you explain to the jury what you actually mean by touch DNA?
It's pretty much what it sounds like. If someone touches an item, their DNA can be left behind. That doesn't mean I'm going to always be able to get a DNA profile. There are several different factors that affect whether I can get a DNA profile. Just because I touched right there, if I take a swab and swab it like I did the glove, I may not see my DNA, but that could be because I washed my hands right before I came in. So there are several different factors with regarding touch DNA, but basically it's just if someone touches something, there's a possibility that I could get their DNA profile.
And what kind of cells are you looking at when you're looking at touch DNA?
When we get DNA profiles from when someone touches something, it's what's called epithelial cells, which is a skin cell, basically.
Just sort of I guess let me kind of ask a question. If I were to, me personally, if I were to put my hands around your neck and then put on a pair of gloves, would you expect to see my DNA on the inside of that glove?
There is a possibility that it would be there. Again, like I just talked about, there's not really any hard and fast rule for touch DNA. Even though we're doing a lot of touch DNA, the research out there doesn't have a hard and fast rule that if you touch something, it's definitely going to transfer. But it is a possibility.
If I were, again, to put my hands around your neck and then put on a pair of gloves, is it possible to find your DNA on the inside of that glove?
Yes, ma'am.
Even though you never touched the inside of the glove?
That's correct.
At that point, would you expect to find a mixture of that DNA?
Yes, ma'am.
And I know you said there's no hard and fast rule. However, let me ask you this. Are you more likely to find that sort of transfer of DNA that I was just discussing when there's more friction involved?
Yes, typically more friction leaves more DNA.
Now, during the time period that you were at the GBI, did you have an occasion to test any known DNA samples that were brought to you by the GBI agents to see if it matched the male DNA on that glove?
Yes, ma'am, I tested several.
When you say several, any idea how many you actually tested?
I would say probably 10 to 20.
Okay. And let me kind of go back. When did you leave the GBI?
I left the GBI, my last day was at the end of April of 2006.
Okay. So, do you know when you first tested this glove?
I tested it October of 2005, October 31st, I believe.
So from the end of October 2005 until, was it April of 2006?
Yes, ma'am.
You tested, you said several, maybe 10 unknown persons to see if they matched.
Yes, ma'am.
Did anybody that you tested match the DNA that was on that glove?
No, ma'am.
And I guess let me be more specific, the male DNA that was on that glove.
No, ma'am.
Did you test any other items in this case to find if there was any DNA present?
I did.
And can you tell us specifically, did you ever test a comforter that was brought to you by the agents in this case?
I did.
Okay. And what did you test on that comforter? What was it that you were testing?
I tested for the presence of blood.
And when you test for the presence of blood on a comforter of this nature, do you actually get the whole comforter
Yes.
from the lab?
Yes.
Do you test the whole comforter?
I test anything when I get an item like that, I've usually requested to test for the presence of blood or semen. There's usually a specific request made. So in this particular case, when a request is made to look for blood, I'm testing anything that looks like blood, a reddish-brown stain, anything like that.
So to show you what's been previously admitted as State's Exhibit 23 in this case. Can you see that on the screen?
Yes.
Can you tell me in particular, this particular item that we're looking at, does that seem consistent with the item that you tested for blood?
To be perfectly honest, I haven't been able to obtain my notes for the comforter, so I don't have any recollection of exactly what it looked like. And without it having the case number there, I can't really answer that.
Understand. So then let me ask you this. So that particular item that you're looking at, can you look at that and with your eyes say, oh yes, that's blood?
No, I can tell you that it's reddish-brown staining, but I would have to perform some chemical tests to actually say whether it's blood or not.
When you test blood, are you able to tell us whether or not it's human blood versus, say, deer blood?
Well, I can test it and tell you whether it's human or non-human.
Okay.
So it's a test for human blood typically, or I guess sometimes we don't actually do the human test anymore. We can go straight forward and perform DNA because that's going to let us know whether it's human or not also.
Okay. Did you do that back in '05, however?
I did not. I did test for the presence of human blood.
Okay. And the comforter that you tested related to this case, can you tell us if it tested positive for the presence of human blood?
Yes, ma'am, it did.
Now, I know you were not there for very long after this case occurred. Do you know if it was ever tested and compared to Tara Grinstead?
I do not.
Just a moment. Back in 2005, when you tested, I'm going back to the blood, when you tested the blood for touch DNA, had you done much work with touch DNA at that time?
No, ma'am.
Do you have any idea how many times you had tested for touch DNA prior to this case?
I actually don't remember any times performing touch DNA because it was something so new. I had tested items, for instance, like a baseball cap left at a robbery scene. I've tested it for what we call wearer DNA, but that's actually more DNA than what a typical touch leaves.
No further questions.
Fox,
Your Honor, we have no questions. Thank you.
all right, you step down.
All right, thank you.
And Judge, would you be free to go?
Any objection, don't you? All right, you're excused. You call your next witness.
State calls Ashley Hinkle. You solemnly swear affirm the testimony you're about to give before the court in this case will be the truth, the whole truth, and nothing but the truth to help you God.
I do.
So tell us your name, please, ma'am.
Ashley Hinkle,
If you'll spell your first and last name for the court, please.
ASHLEY-HINKLE.
And Ms. Hinkle, how are you employed?
I'm employed at the Georgia Bureau of Investigation in the Forensic Biology section in Decatur, Georgia.
How long have you been employed with the Georgia Bureau of Investigation in the Forensic Biology section?
Approximately 12 and a half years.
Now, Ms. Hinkle, if you will, give the jury the benefit of your training and experience and allow you to do your job as a forensic biologist.
I have a Bachelor's of Science in Biology from East Carolina University and a Master's of Science in Forensic Science from Marshall University, as well as extensive in-house training at GBI and the National Forensic Science Technology Center in Largo, Florida.
Now, with regard to being a forensic biologist, if you will, tell us what you do as a forensic biologist.
Primarily, my job as a forensic biologist is to examine evidence for the presence of bodily fluids. If those are found, I attempt to obtain a DNA profile, and I compare that profile to reference samples that have been submitted from individuals.
Now, when we talk about bodily fluids, when we say bodily fluids, what kind of bodily fluids are you looking for?
Blood, semen, or saliva. We also test contact DNA.
Now, with regard to, we talk about DNA, what exactly is DNA?
DNA stands for deoxyribonucleic acid. It's found throughout our bodies in what's called our cells. It gives us unique characteristics such as the color of our eyes or the color of our hair. No two people have the same DNA except for identical siblings, and we get half from our mother and half from our father.
All right. Now, with regard to the testing and training that you've done there at the crime lab, have you testified as an expert in the field of forensic biology before today?
Yes, I have.
And do you recall how many times you've done that, Ms. Hinkle?
Approximately 140.
All right. Now, Judge, this time we would tender Ms. Hinkle as an expert in forensic biology.
All right, she's admitted as an expert.
Now, Ms. Hinkle, what kind of testing do you do for looking for something for DNA and then the comparison of that DNA? What do you do? What kind of testing do you do?
Do you want me to take you through the steps?
Yes, ma'am, please. Sorry.
Yes.
Bad question. My fault.
We perform testing, DNA typing. We take a small amount of a sample, we put it in a tube. We add liquids to separate the DNA from the other biological material that's present. We use a robot to assist in this step. We then quantitate the amount of DNA that's present to tell us if we actually obtain DNA from the item. This DNA is then copied on an instrument, the PCR. We make copies along the DNA strand. These copies are separated on an instrument called a genetic analyzer. And from the genetic analyzer, we actually get the data we've used in a software program, and that provides us a DNA profile. Then we physically have to compare an unknown sample or sample from evidence to reference samples.
Now, with regard to this case involving the disappearance of Ms. Tara Grinstead, were you asked to look at items of evidence in this case for comparison for DNA purposes?
Yes, I was. I started on this case around 2010.
Right. Now, with regard to that, Ms. Hinkle, I'm going to show you. Show you several photographs, that being States Exhibits 28, 29, and 91. And ask if you'll look at those, please, ma'am. Are you familiar with what's shown in those photographs?
I haven't actually seen these photos before.
Okay. Do you recognize, with regard to items of evidence, do they have a specific case number associated with the case?
Yes, I do recognize the GBI case number 2005-1025070.
Okay. And with regard to that particular case number, is that associated to a particular case involving the disappearance of Tara Grinstead?
Yes, sir.
With regard to that, did you test a glove with regard to Ms. Grinstead?
Yes, I did.
All right. Now, with regard to the glove is that similar to the glove that's shown in those photographs?
Yes, I did.
Okay.
Yes.
Now, with regard to that particular glove, what were you asked to do originally with your testing regarding results from Ms. Ellis or Ms. Hopgood Ellison that used to work at the lab when you got this? And I'll refer you, so we're clear, I know you get a bunch of reports. So I'm going to refer you to your report, Ms. Hinkle, dated June the 10th of 2015. Okay? What were you asked to do with regard to this item, with regard to the results from Ms. Hopgood or Ellison's report back from them?
At that time, it was requested that we send off DNA to testing for what's called phenotype testing. Phenotype testing is different than what we do at GBI, but it helps to give an image of who the DNA belongs to. They use certain factors that tell you the color of the hair, whether someone has a cleft chin, ethnicity of an individual. So that was requested. I had to actually retest the glove because they needed a sample that had around 90% from the unknown male. And the sample that had been previously tested by Ms. Hobgood it was about a 50-50 mixture.
Okay. And so when you retested the sample, do you recall what portion of the glove it was that you retested?
I tested a portion of the glove. You
Put it on the screen for you if you'd like.
can see the middle part around the knuckles is what,
Take position number 28. Do you see that on the photograph there?
yes, that middle area is around the knuckles. Or the top of the palm of the hand. It's not really the knuckles, it's the top of the palm of the hand. It's what we were provided for DNA testing.
Okay, so just so we're clear, there are items up here that look like fingertips. Is
Yep.
that the item you tested?
No,
And regarding, there's a long strip at the bottom. Is that the item you tested?
no, sir.
Now, this item here, is that the item you're referring to?
Yes, that's what I tested. I successfully obtained a profile from. Those other items, they actually were tested, but it was after the late prints processing, and I wasn't able to obtain any DNA for the phenotype testing.
In regard to the testing that you performed, with regard to that particular area of this glove, did you perform those tests as you previously described for us?
Yes.
And did you perform those in the manner in which your protocol and procedures require?
Yes, I did.
And what were you able to determine from your testing with regard to that area of the glove?
I actually tested it separately. I took swabbings from what was the inner portion of the glove and the outer portion of the glove. These may not actually be the inner and outer portion of the glove. It was just as I received it. From the inner portion, I obtained a DNA profile that was of 2 individuals, and from the outer, I obtained a DNA profile that was from 3 individuals.
Now, with regard to the inner profile that you talked about, you said that you had a profile of 2 individuals. Is that right?
Correct.
Now, with regard to your testing, were you able to, did you have a known sample for your testing from Ms. Tara Grinstead?
Yes, I did. At that time, we used Ms. Grinstead's toothbrush.
Now, with regard to the known sample from Tara Grinstead, did you also, and this may be in a different report, at some time have, like I believe your report is July of 2016, did you receive a buccal swab sample of known DNA from a person known as Bo Dukes?
Yes, I did.
All right. Now, with regard to the 2 individuals that were found on that inner portion of the glove in your testing, did you make any determination of whether that was that individual, with regard to those 2 individuals, were you able to make a determination as to Mr. Bodukes?
It did not match Mr. Bodukes.
And with regard to Mrs. Tara Grinstead, with regard to the known sample from her toothbrush, what did your test and findings indicate?
From the known sample from the toothbrush, there was a partial profile from the inside of the glove. It matched Ms. Grinstead, and approximately 1 in 900 individuals would also have that profile.
Now, with regard to later, let me say, I think it's directed to your report, which is same one, sorry, same one. With regard to your report, and the report date is February 24th of 2017. At this time, did you receive a known buccal swab or DNA sample from someone known as Ryan Alexander Duke?
Yes, I did.
Now, with regard to the known sample of Ryan Alexander Duke, did you compare that to the known profile from this portion of the glove, the other individual that was there?
Yes, I did.
And if you will, did you perform your testing according to the procedures you've already described for us?
Yes, sir.
And if you will, give us the results of your testing against the known DNA sample of Ryan Alexander Duke and the other individual on this glove besides Ms. Grinstead.
The inner piece of glove, the primary profile matched the DNA profile of Ryan Alexander Duke.
With regard to the profile of Ryan Alexander Duke, were you able to determine a frequency with regard to that?
Yeah, the frequency of finding this profile randomly in the general population would be 1 in 300 quadrillion individuals.
Now, I'm not good at numbers, and I'll just tell you that up front, but when you say quadrillion, what does that mean?
Well, quadrillion is 15 zeros, and since it's 300, you would add 2 more, so it would be 17 zeros. There's only about 7 billion people in the population of the Earth, and 7 billion, that's 9 zeros.
So this is more than the population of anyone or the number of people on planet Earth?
Correct.
Now, Ms. Hinkle, are you familiar with when I say something called touch DNA? Are you familiar with touch DNA?
Yes, I am.
And when I say touch DNA, can you tell me what I'm talking about?
Touch DNA, another name for it is contact DNA. It's not from biological fluids such as blood, semen, or saliva. It's mainly skin cells. If I were to touch this area right here, I could leave some of my DNA, or I could actually not leave some of my DNA.
Sorry.
But this would be a touch item as far as evidence.
Now, with regard to touch DNA, are you familiar with, have you testified before about touch DNA and admitting that into evidence?
Yes, I have.
Now, Ms. Hinkle, through your time at the crime lab, did y'all also have a comforter that you tested for the presence of blood? Not that you specifically did it, but was there a comforter there that was tested for blood as well?
Yes, there was.
All right. And was there another analyst there at the lab that tested that blood for Ms. Grinstead's blood? Is that right?
It was another analyst, yes.
Okay. And it was found to be Ms. Grinstead's blood?
Yes. And can I add that actually at the time of the Parabon testing, we did need a known sample of Ms. Grinstead, so it was a cutting from a comforter that we used in order to provide a known sample, because by that time, there wasn't enough DNA left on the toothbrush.
Okay. And with regard to that specific sample of being Ms. Grinstead's blood on the comforter, and I apologize for this question, I don't know how else to ask it. Are you able to tell the difference of the source of the blood or the kind of blood that it is when you do that kind of testing?
No, I can't tell if it's blood from a struggle, if it's blood from a nosebleed, or if it's menstrual blood.
I don't have anything further of Ms. Hinkle.
Mr. Fox?
Listen, just to reiterate, when you tested male DNA on the blood, it did not match Bowden's.
Correct.
Thank you.
Anything from? All right, you can sit down.
Thank you, sir.